One of the leading factors of global warming is the building up of some gases known as greenhouse gases. Examples of gases include carbon dioxide, methane, nitrous oxide, and chlorofluorocarbons. These gases inhibit the outgoing radiations from the Earth and also upset the Earth's heat balance. About 60% of methane expelled in the atmosphere is man-made, sourced from landfills, the petroleum industry, agriculture, and many more. But did you know that there exists a diverse group of microorganisms that can use these one-carbon compounds like methanol or methane as the carbon source for their growth? So the question is, how do microbes metabolize methane? There are several alternative pathways that provide energy aside from conventional glycolysis. First off, we have aerobic hydrocarbon oxidation. Aerobic hydrocarbon oxidation is a process where microorganisms use hydrocarbons and convert them to energy. Through stepwise oxidation, hydrocarbons are oxidized by oxygen to produce water. A notable example of microorganisms that use hydrocarbons are Cladosporium resinae and Pseudomonas aeruginosa, also commonly called humbugs, and are usually found in jet fuel. Next, we have the pentose phosphate shunt. It's an alternative energy-producing process that breaks down glucose 6-phosphate into NADPH and pentoses, which will be further used in downstream biological processes. There are two stages: first, the oxidative phase, and second, the non-oxidative phase. The oxidative phase is the phase where NADP+ and NADPH are produced through the conversion process of glucose 6-phosphate to ribulose 5-phosphate. Their non-oxidative phase constitutes the synthesis of 5-carbon sugars. In this phase, ribulose 5-phosphate can isomerize to ribose 5-phosphate and vice versa. Organic acid metabolism is divided into two: formate metabolism and fatty acid metabolism. Formate metabolism is important in the breakdown of C1 compounds like methanol. The catalysis of formate allows for the donation of electrons to substrates such as NAD+. Single carbon compounds are converted into formaldehyde, followed by its oxidation to formate through the enzyme formaldehyde dehydrogenase. Fatty acid metabolism uses the beta oxidation pathway, which breaks down the fatty acid chains into acetyl-CoA. Fatty acid metabolism can be summarized in the following steps. First, fatty acids are converted to enoyl-CoA. Next, enoyl-CoA is converted to 3-hydroxyl-CoA. 3-hydroxyacyl-CoA is converted into 3-ketoacyl-CoA. And lastly, 3-ketoacyl-CoA is diolated to produce acetyl-CoA and a derivative of the original fatty acid chain, which has been reduced by 2 carbons. Next, we have lipid metabolism. Lipids are metabolized from microorganisms' primary source of energy, and it is done through beta oxidation. In beta oxidation, fatty acids and lipid derivatives are being broken down, in which 2 carbon fragments are removed from the carboxyl group end of the fatty acid. This process can be summarized into the following steps: activation, oxidation, hydration, oxidation, and cleavage. For the main topic, we are going to talk about C1 metabolism. 1-carbon compounds at all oxidation levels can be found in nature, including methane, methanol, methylated amines, dimethyl sulfide, formate, and chlorinated methanes. Methanol is formed by the hydrolysis of plant methyl esters and ether, such as pectin and linen. Methylated amines are produced by microbial degradation of choline derivatives present in plant membrane material and animal tissue. Dimethyl sulfide arises from the cleavage of dimethyl sulfoneopropionate, a product of acyl sulfur metabolism. Formate is a major end product of mixed acid fermentation, and carbon dioxide is present in the atmosphere as carbonates in natural waters and soil. Finally, chlorinated methanes are frequently used as solvents and are produced in enormous quantities in the industry. Microbes that use C1 compounds or any multi-carbon compounds that lack carbon bonds as their sole source of energy for their overall growth are known as methylotrophs. Yeast species able to use methanol as carbon and energy source belong to genera Pichia and Candida. These organisms derive methanol from the methoxy groups present in wood lignin or fruit pectin. Many strains of methylotrophic bacteria are typically obligate methylotrophs. Those methylotrophs able to utilize methane as sole carbon and energy source generally are referred to as methanotrophs, which will be discussed later. Generally, methylotrophs aerobically oxidize C1 compounds in order to yield formaldehyde. Then, this formaldehyde can either be dissimilated to carbon dioxide or assimilated into biomass, which the cell can use as a source of energy and carbon for growth. The combined action of these dissimilatory and assimilatory pathways result in the synthesis of a metabolite containing a C3 skeleton. Once such a compound has been produced, synthesis of cell constituents further proceeds via the intermediary metabolism. Microorganisms growing on C1 compounds possess a special set of enzymes for the above-mentioned activities. For example, in methanol oxidation, methylotrophic yeasts employ an alcohol oxidase which contains FAD as a prosthetic group and is localized in peroxisomes. Whereas methanol dehydrogenase is involved in the oxidation of methanol to formaldehyde in the periplasm of gram-negative bacteria. There are 4 different pathways which affect the synthesis of compounds containing carbon-carbon bonds from C1 units. These pathways are the Calvin cycle or ribulose bisphosphate pathway of carbon dioxide fixation, the ribulose monophosphate cycle of formaldehyde fixation, the cellulose monophosphate cycle of formaldehyde fixation, and the serine pathway in which both formaldehyde and carbon dioxide are fixed. The serine pathway involves carboxylic acids and amino acids as intermediate, whereas the other 3 involve carbohydrate intermediates. The ribulose monophosphate or rump cycle is only found in yeasts. It is also known as the Calvin cycle. In the ribulose bisphosphate pathway, cell carbon is assimilated in the level of the carbon dioxide produced by oxidation of the reduced 1-carbon substrate. This pathway consists of 3 main parts: fixation, reduction, rearrangement. Fixation. The fixation part involves the carboxylation of 3 molecules of ribulose biphosphate to 6 molecules of 3-phosphoglycerate, which are then phosphorylated to 1,3-diphosphoglycerate. Reduction. These are then reduced to 6 molecules of glyceraldehyde 3-phosphate, one of which is the end product of the pathway. Rearrangement. The third part consists of rearrangement reaction, which regenerates 3 molecules of ribulose biphosphate. At least 2 variants of the cycle occur which differ in their rearrangement reactions: pseudoheptulose bisphosphatase and transaldolase. The ribulose monophosphate pathway is similar to the ribulose bisphosphate pathway, except that the reduced carbon substrate is oxidized only to formaldehyde prior to assimilation. In other words, all cell carbon is assimilated at the level of formaldehyde, produced by the oxidation of reduced C1 compounds. In this pathway, 3 carbon compounds, or pyruvate, are synthesized from 3 moles of formaldehyde. The main steps are fixation, cleavage, and rearrangement. Fixation. In this part, formaldehyde is condensed by an alcohol condensation reaction with ribulose 5-phosphate to give hexulose phosphate. Then this hexulose phosphate is isomerized to fructose 6-phosphate. Cleavage. Here, 1 molecule of fructose 6-phosphate is converted to either 2-keto-3-deoxy 6-phosphogluconate or to bisphosphate, which are then cleaved by aldolases to glyceraldehyde 3-phosphate, plus the product to the pathway, which is either pyruvate or dihydroxyacetone phosphate. In the 2-keto-3-deoxy-6-phosphogluconate aldolase or transaldolase variant, which occurs predominantly in obligate methylotrophs, fructose 6-phosphate is converted to 2-keto-3-deoxy-6-phosphogluconate or KDPG by several enzymes, and then cleaved by KDPGA to glucose 6-phosphate and pyruvate. However, only one NADPH can be generated in this variant. The second variant is the fructose bisphosphate aldolase variant or the FBP aldolase, in which the fructose 6-phosphate is converted into fructose 1,6-bisphosphate and subsequently cleaved by FBP aldolase to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, which can be further converted into pyruvate, generating 1 NADPH and 2 ATP. Rearrangement. Lastly, glyceraldehyde phosphate and FMP undergo a series of reactions which lead to regeneration of ribulose 5-phosphate. Remember that there are two possible rearrangement variants, cytoheptulose 1,7-bisphosphatase and transaldolase, and two possible cleavage enzymes. There are a total of 4 potential variants for the RUMP pathway. However, the last few steps are the same in two variants, which are glyceraldehyde 3-phosphate and cytoheptulose 7-phosphate, rearranged by transketolase to 2 5-carbon sugar phosphates, cellulose 5-phosphate and ribose 5-phosphate, which is further converted to ribulose 5-phosphate and completed the regeneration pathway. Formaldehyde is firstly combined with cellulose 5-phosphate by peroxisomal enzyme DAS, forming dihydroxyacetone DHA and glyceraldehyde 3-phosphate GAP. Then, DHA and GAP will be further assimilated in the cytosol. DHA or dihydroxyacetone is phosphorylated to form dihydroxyacetone phosphate or DHAP, which can be condensed with GAP to form fructose 1,6-bisphosphate. In the first reaction, formaldehyde reacts with glycine to form L-serine. L-serine is then transaminated with glyosylate as the amino group acceptor and produces hydroxypyruvate and glycine, in which this glycine can also be recycled to serve as a substrate for serine hydroxymethyltransferase. Next, hydroxypyruvate is reduced to D-glycerate, followed by phosphorylation to produce 2-phospho-D-glycerate. To summarize, assimilation converts C1 compounds into C3 compounds. Listed in the table are the C1 compounds fixed in each pathway and the final products. Next, we will discuss the other fate of formaldehyde, which is dissimilation to CO2. Formaldehyde dissimilation is ubiquitous in living organisms, as formaldehyde is commonly produced from various demethylation reactions in cells. Microorganisms possess a variety of pathways to fulfill formaldehyde oxidizing reactions. In yeast, dissimilation may occur via the cellulose monophosphate pathway, although they contain enzymes catalyzing the direct oxidation of methanol to carbon dioxide. Let's recap what we have learned so far. We learned that C1 compounds include methane, methanol, methylated amines, dimethyl sulfide, formate, and chlorinated methanes. We also learned that those organisms that can utilize C1 compounds as their source of energy for growth are called methylotrophs. They do this by first oxidizing C1 compound to formaldehyde using special set of enzymes. Then, formaldehyde either undergoes assimilation through one of the 4 pathways, namely ribulose bisphosphate pathway, ribulose monophosphate pathway, cellulose monophosphate pathway, and serine pathway to produce C3 compounds that are converted cell constituents via intermediary metabolism, or formaldehyde undergoes dissimilation to CO2. However, methane oxidizing bacteria encompass other organisms that are not regarded as sole methanotrophs. For this reason, methane oxidizing bacteria have been separated into 4 subgroups: 2 methane assimilating bacteria or MAB groups, the methanotrophs, and the 2 autotrophic ammonia oxidizing bacteria or the AAOB. Methanotrophs oxidize methane by first initiating reduction of an oxygen atom to hydrogen peroxide and transformation of methane to methanol using methane monooxygenases. The two types of MMOs, or methane monooxygenases, are soluble methane monooxygenases, which is found in the cell cytoplasm, and particulate methane monooxygenases, which is found in the cell membrane. Type 1 and Type 10 methanotrophs utilize the RUMP pathway or the ribulose monophosphate pathway, while the Type 2 methanotrophs make use of the serine pathway. Under aerobic conditions, methanotrophs combine oxygen and methane to form formaldehyde, which is then incorporated into organic compounds via the serine pathway or the ribulose monophosphate pathway, which releases carbon dioxide. Type 1 and Type 10 methanotrophs are part of the gamma proteobacteria, while Type 2 methanotrophs are part of the alpha proteobacteria. Under anaerobic conditions, methanotrophs use different electron acceptors for methane oxidation. This can happen in anaerobic habitats such as marine or lake sediments, oxygen minimum zone, anaerobic water columns, and rice paddies and soils. Some specific methanotrophs can reduce nitrate or nitrite and couple that to methane oxidation. Methane is the major constituent of natural gas and is also present in many coal formations. Methanotrophs are of special interest to researchers studying global warming because they play a significant role in the global methane budget by reducing the amount of methane emitted to the atmosphere, which is far more potent than carbon dioxide from being released into the atmosphere. The amount of methane that escapes to the atmosphere is the difference between the amount produced from the various sources and the amount consumed by the methanotrophs and anaerobic methane oxidizing bacteria. Some methane is consumed from the atmosphere by methanotrophs in soils of forests, grasslands, and other unsaturated soils, which are the major terrestrial sinks for atmospheric methane. Here are the following sources and sinks of atmospheric methane. Wetlands are considered to be the major source of atmospheric methane, and the major influences on methane emissions in tundra soils are vegetation and soil moisture content. Next, methane production in flooded rice paddies is approximately 575 teragram a year, but that amount escaping to the atmosphere is only about 100 teragram a year. Freshwater lakes account for only a small portion of the total atmospheric methane budget. Oceans are a minor source of atmospheric methane, where the highest concentrations can be found in near surface waters. Marine waters near hydrothermal vents located at mid-oceanic spreading centers at depths of 2,000 to 3,000 meters have high levels of methane and are sites of aerobic methane oxidation. Next, some soils serve as atmospheric methane sinks, while aerobic soils serve as biofilters for produced methane of anaerobic soils and sediments. Very high concentration of methane oxidation have been observed in landfills covered by soil, and they are estimated to produce about 30 to 70 teragram of atmospheric methane yearly. Interestingly, significant amounts of methane can be found from ruminants, coal mining, oil and gas recovery, and gas transport. After talking about all these processes, we question, how are these concepts applied in real life? And what are the recent advancements that can potentially impact our lives? We already know that methanotrophs can convert methane through a metabolic process known as C1 metabolism and convert it to biomass. Aside from this, it has also been shown that intermediates of C1 metabolism can be used as backbone to produce more complex chemicals such as vitamins, cosmetics, biofuels, and lipases, with many industrial uses such as soaps. These are only a part of the variety of other applications of methanotrophs, and since science has come a long way, let's look at the advancements of ethanol-based industrial biotechnology. Framework shows key factors and future works of methanol-based industrial biotechnology. Let's focus on three things. 1. Synthesis of amino acids. 2. Biological conversion of methane and methanol for manufacturing of products. And 3. Sustainable methane from anaerobic digestion. Let's focus on the first one. So amino acid fermentative processes using methanol media are being studied and explored because of its opportunity to reduce feedstock prices and relieve the high burden of wastewater treatment of currently employed large-scale fermentations with molasses. Aside from the synthesis of amino acids, according to Backer and Whitman in 2015, microbial conversion of methane and methanol allows for the economic bioproduction of different industrial products and commodity chemicals for health and nutrition, especially since methane and methanol are already chemically transformed into compounds used for the manufacture of materials such as lead and LCD devices presently. Lastly, according to Abbasi and his co-authors in 2012, in various industries such as wastewater treatment, brewing, and agricultural and livestock residue, microbial methane production and capture are an established technology utilized for the treatment of organic waste streams, and the methane produced during anaerobic digestion is seen and referred to as a sustainable supply derived from a renewable source. Methanotrophs can also be used in biogas production. Biogas, which is primarily composed of methane and carbon dioxide, with smaller amounts of hydrogen sulfide and other gases, is produced by the breakdown of organic waste through anaerobic processes. With the use of methanogens and other anaerobic organisms, organic materials are digested in a closed system called an anaerobic digester, biodigester, or a bioreactor. Biogas can be used to produce electrical thermal energy through fuel cells or combustion engines, where biogas is converted into mechanical energy that powers a generator producing electricity. Biogas is produced in a biogas plant, which contains 5 major sections. 1. The mixing tank, 2. The inlet chamber, 3. The digester, 4. The outlet chamber, and 5. The outflow tank. The mixing tank is where organic waste is mixed with water. This forms the slurry. Among the different types of organic waste, animal dung is the most commonly used since methanogens in dung have more potential in producing biogas than others. The slurry is then digested into the digester through the inlet chamber. Once the digester is partially filled with the slurry, the plant is left unused for about 2 months. This process is important because this is the time the microbes do their job in producing biogas by decomposing or fermenting the biomass in the presence of water. As the pressure increases inside the digester, biogas is collected and the spent slurry is moved into the outlet chamber. Once the spent slurry overflows into the overflow tank, it can manually be removed and used as manure for plants. And that concludes our presentation. We hope you have learned a lot from us, from the C1 metabolism to the methanotrophs and to their applications in the industry.